Moderna has a patent on the use of RNA for vaccines. It happens to be patent number 2019-024-0317A1. And in that, Moderna explicitly acknowledges that that RNA is superior to DNA for vaccine purposes because problems, including the possibility of insertional immunogenesis, which could lead to the activation of oncogenes or the inhibition of tumor suppressor genes. Okay, so FDA says they're not aware of any concerns, but Moderna, in its own patent, lays out exactly the same concerns that exist about DNA and insertional immunogenesis and genotoxicity. Okay, so Moderna knows it.
Was the DNA a contaminant?
It is a contaminant. It is left in because the way they make it in process 2 is they purify circular DNA from bacteria. They make the RNA. RNA makes DNA. I'm sorry, DNA makes RNA. RNA makes protein. Okay, so they use DNA to make the RNA, and then they degrade the DNA, and then they have to purify the degraded DNA away from the RNA. And the process that they're using is apparently not very good. And so they still, and this was just revealed. It was just detected because some scientists, both in the US and in Canada, managed to get their hands on some unopened vials with clear chain of custody, sample those vials, and these are people who their business is to do deep sequencing on samples and rebuild information about whatever DNA they find on that. So they were just doing the technology that they know how to do in their business, that they're experts in, and they found, lo and behold, that there were these large numbers of DNA fragments in this RNA preparation, and they could apply standard tools that they have to reconstruct what those circular plasmid DNAs look like. Okay? None of this was disclosed to the public. And it appears that the documentation is that there are certain sequences present that are normally not allowed in anything that's going to go into humans, not the least of which is an antibiotic resistance gene. Okay? So this is going, these are going into our bodies, including an antibiotic resistance gene for kanamycin or neomycin. Normally, you're not allowed to do that if it's a DNA engineered for human use, okay? And they include these sequences from simian virus 40, not the whole virus, but highly active promoter sequences, which is exactly the thing that the FDA in their older regulations said must be avoided because it confers even more risk for insertional immunogenesis. And what appears is that Pfizer, when they provided these documents to the FDA and to the EMA and to Health Canada, they took these standard plasmid maps that are generated through programs that we all have access to, and they deleted the little notation that said there's SV40 sequences in there. And the FDA, apparently out of, I can only think, incompetence, didn't take the raw DNA sequences, reconstruct those plasmid maps, and look at them themselves. They just took for granted what Pfizer had given them. And now this has all come out because of these researchers that did functionally something akin to the lot release testing that is not happening.
So there was probably somebody at Pfizer who knew what they were doing.
Absolutely.
Okay.
And Moderna.
And then what are the specific downstream diseases or bad outcomes that come from having these?
Anything that is associated with DNA damage.
For instance, cancer?
Birth defects and cancer being the notable ones.
Okay.